Welcome to BioTales, where we share the fascinating journeys behind biotech's biggest breakthroughs. Today, we've got a truly exciting story, really hot off the presses, actually. Just a few weeks ago, we talked about Capstan Therapeutics and their massive $2.1 billion acquisition by AbbVie for their in vivo CAR T technology. Now, almost immediately after that, another major deal is dropped in the very same groundbreaking space. We're talking about Interius BioTherapeutics. They're a company with a shared vision to engineer CAR T cells directly inside a patient's body. And they've just been acquired by Kite Pharma, that's the cell therapy arm of Gilead Sciences, for $350 million. So two innovative startups, two big acquisitions, one truly audacious idea. Can we make these powerful CAR T therapies inside the patient, not in some distant lab?
What's really fascinating here is that these acquisitions coming so close together really highlight a massive shift in how the industry is thinking about advanced cell therapies. It's not just about the money, though. That's significant. It's really about a potential paradigm shift, one that could make these life-saving treatments accessible to so many more people.
It's an incredible journey we're going to unpack for Interius BioTherapeutics. It starts with this almost frustration from an oncologist seeing patients wait, then navigating skeptical investors, achieving these amazing preclinical results, launching global trials, and, well, ultimately, this big acquisition.
Yeah, it's a fantastic story of how a seemingly impossible idea can really start to change everything.
So to really get why Interius is such a big deal, maybe we should quickly touch on traditional CAR T again.
Good idea. So traditional, or ex vivo, CAR T therapy. It's been a genuine game changer, no doubt, especially for certain blood cancers. Some people call it a living drug. And the process, like you said, involves taking a patient's own T cells out of their body. Then, in a specialized lab, they're genetically engineered to recognize and attack cancer cells. After that, you have to grow billions of these newly weaponized cells, and then finally infuse them back into the patient. When it works, it can be truly miraculous.
Miraculous, definitely, but also incredibly difficult, right? The whole process is so complex.
It really is. You're looking at weeks, sometimes many weeks, just for the manufacturing.
And during that time, the patient is just waiting, often very sick, needing harsh chemotherapy, first the lymphodepletion, which just wipes out their immune system.
Exactly. That chemo makes them incredibly vulnerable. And then there's the cost. We're talking six figures, often very high six figures.
I remember hearing from doctors, it's heartbreaking. They watch patients get sicker, sometimes even, not making it, just waiting for their cells to be ready.
These aren't just small problems. They're massive hurdles. The time, the cost, the logistics. It means a lot of patients who could potentially benefit just can't access the therapy. Maybe it's the cost, maybe it's where they live.
And it was seeing all this day in and day out that really got to Dr. Saar Gill at the University of Pennsylvania.
Right. He's an oncologist or researcher. He was living this reality. Even though he co-founded another company, Charisma Therapeutics, working on the CAR macrophages, he kept coming back to this fundamental issue with the ex vivo approach. Taking cells out, doing stuff to them, putting them back in. It felt cumbersome.
So he started asking this really radical question, probably sounded crazy at the time. What if we didn't need the lab step at all? What if we could just inject something, a vector, that finds the T cells in the body and reprograms them right there, in situ?
That's a massive conceptual leap. I mean, for years, gene therapy was about targeting cells you could easily get to, or taking them out first. The idea of engineering a whole population of circulating immune cells directly inside a living person, the control needed, the specificity, it seems almost like science fiction.
Until Gill started to piece together how it might be possible.
Exactly. But the need for it was undeniable. A faster, cheaper, more accessible CAR T that could really bring this powerful therapy to so many more people.
So Gill's frustration became kind of an obsession, it sounds like. He dug into the research, experimented in the lab, laying the groundwork for what would become Interius.
That core idea, engineering T cells within the body. But, an idea, even a brilliant one, needs more. It needs a team. It needs money.
Then he met with Bruce Peacock.
Right. Bruce Peacock is this really seasoned biotech entrepreneur. He's got a track record of building companies and, frankly, knowing a good idea when he sees one. People say when he met Saar Gill, probably around 2019, he was just instantly grabbed by Gill's vision and that simple question, why not do it inside?
So they started mapping things out. They needed a name.
And they landed on Interius. It's Latin for within. Pretty perfect, right? Captures the whole mission.
Totally. And they knew they needed more than just the science and the vision guy. They needed operational strength, investor credibility.
Which led them to Phil Johnson. He's a Physician scientist, really deep experience with viral vectors from previous roles. He eventually became CEO. He brought that crucial mix of scientific understanding, business sense, and importantly, the kind of background that gives investors confidence.
So the core team starts to form. They set up shop at Pennovation Works, that incubator space at Penn.
Yeah, those must have been intense early days. Gill and Peacock putting together the pitch deck, focusing on retargeting lentiviral vectors. These are the viruses often used in traditional ex vivo gene therapy, but the idea was to make them super specific, only hitting certain T cells and NK cells in the body.
And they managed to pull together some early money.
They did. About $1.5 million in seed funding. Came from, the usual early sources, friends, family, an early investor called Tellus Bioventures, plus support from Penn itself. Just enough to get started, to try and generate that first bit of really crucial data.
But it wasn't easy convincing people, right? I imagine there was a lot of skepticism.
Absolutely. You pitch injecting a lentivirus, which integrates into the genome directly into the bloodstream. A lot of investors were like, whoa, hold on. How do you control where it goes? What are the risks? Will regulators ever approve something like that? There were major doubts.
But they found people who believed. And Gill was apparently juggling patient care with lab work. Really dedicated.
And with that seed money, they ran those first absolutely critical experiments. Can we actually target just T cells?
How did they do that?
Well, the standard lentivirus uses an envelope protein called VSVG, which lets it infect lots of different cell types. They basically swap that out. They engineered a new envelope protein that included a part designed to bind specifically to CD7, a marker found on T cell. In the lab, in Petri dishes, they added a green fluorescent protein marker to the vector, and they saw the T cells light up green. But other cell types stayed dark. That was the first real, tangible sign. Maybe, just maybe, this impossible idea was actually possible.
Okay, let's dive into that science a bit more, because that's the real magic here, isn't it? Making that vector super specific.
It really is the core innovation. Like we said, the standard VSVG envelope is way too broad for in vivo usage. You infect cells all over the body. Not good.
So Interius needed a precision-guided missile, essentially.
Exactly. And their solution was this engineered envelope protein they called a fusogen. Think of it like a key designed for only one very specific lock. It has two main parts. One part is like an antibody fragment that's the key part that recognizes and binds only to a specific protein on the target cell surface. For them, that target was CD7.
And CD7 is mostly just on T cells and NK cells.
Primarily, yes. Crucially, it's not really found on B cells or other important tissues. So targeting CD7 gives you that specificity. The other part of the fusogen is the fusion domain. That's what actually lets the virus enter the cell after it binds.
Clever. Was it hard finding the right CD7 binder?
Yeah, that was apparently a challenge. They needed something that worked across species for preclinical testing, so they ended up re-engineering an older antibody to fit their need.
Results in testing.
They were pretty stunning. The specificity was incredible, over 99.9% for T cells, barely touching anything else. Then they moved into humanized mice. These are mice with a human immune system. A single injection of their vector carrying a CAR gene.
What happened?
It generated CAR T cells inside the mouse. And those cells went to work, wiped out the target B cells, even eliminated tumors. That had to be the real aha moment. Proof that you could literally turn T cells into targeted killers, right there, inside a living being.
Wow. And they stuck with lentiviruses specifically because they integrate.
Yes, that was a key strategic choice. For a therapy like CAR T, you want it to last. You want the cancer-fighting gene to stick around as the T cells divide and multiply. Integration into the cell's genome provides that durability. Other approaches, maybe using certain nanoparticles or non-integrating viruses, might not give you that long-term effect.
But integration has risks, right? Like causing other mutations.
It does, and that's a major safety focus. Interius tackled this in a few ways. They use what are called self-inactivating long-terminal repeats. Think of them as genetic safety switches that help control the activity of the integrated gene and reduce the risk of it activating nearby cancer genes. Plus, they carefully controlled the dose to limit the number of integrations per cell and did extensive studies looking at exactly where the virus was integrating to assess the risk.
Okay, so by late 2021, they had strong data. Specific, potent, seemed safe in animals, and they could make this stuff.
Exactly. They had the package. Specific delivery, powerful effect, persistent cells. They were ready to take the next big step, raising serious capital.
Which brings us to early 2021. The pandemic is still raging, but Interius is gearing up for its Series A financing.
Right. Dr. Gill, Bruce Peacock, Phil Johnson, they're hitting the virtual pavement. Their pitch is powerful. We can make CAR-T faster, better, and way cheaper.
And the timing couldn't have been better, really.
No, it was perfect. Investors had seen the huge successes of the first wave of ex vivo CAR-T companies like Kite, ironically, and Novartis Juno. But they also knew all about the limitations we discussed, the cost, the time, the complexity. So the idea of an in vivo solution was incredibly appealing.
And it landed. In May 2021, they announced the Series A.
A huge one, $76 million, and it was oversubscribed. They actually had to turn some investors away. It was co-led by Cormorant Asset Management and Fairmount Funds. But look at the syndicate: Bain Capital Life Sciences, Pfizer Ventures, RA Capital, Longwood Fund. These are top-tier biotech investors.
That's a massive stamp of approval. More than just money, it's validation.
Totally. It showed the market really believed this in vivo thing wasn't just a pipe dream anymore. It was a serious contender.
And they put that money straight to work, right? No messing around.
Nope. They scaled up fast, expanded the team, built out proper manufacturing capabilities, started moving towards GMP good manufacturing practice. That's the quality standard you need for human trials.
So things are moving quickly now.
Very quickly. By late 2022, they've got manufacturing humming and key patents filed on the CD7 targeting tech. Fast forward to late 2023, they complete the crucial non-human primate studies. These are the closest thing to humans you can test in.
And those look good.
Yeah. Reportedly show the expected B-cell depletion, which is what the CAR T cells are supposed to do, but without the really dangerous side effects like severe cytokine release syndrome or neurotoxicity that sometimes happen with traditional CAR T. They're also deep in discussions with regulators, figuring out the path to the clinic.
Okay, preclinical data looks great, money in the bank. Next stop, human trials. Big decision time. Where do you start?
Right. The U.S. FDA path is rigorous, can take time. Interius wanted to get moving, get that first human safety data as quickly as possible. So they made a strategic choice: Australia.
Why Australia?
Well, CEO Phil Johnson explained it later. Australia has a reputation for pretty efficient regulatory reviews for early-phase trials. Plus, they offer attractive R&D tax incentives. And critically, they have really top-notch clinical sites and researchers. They found a key partner in Dr. Michael Dickinson at the Peter MacCallum Cancer Centre in Melbourne. He really believed in the approach.
So start in Australia, get that initial safety data fast, and then use that to support applications in the US and Europe later.
Exactly the strategy. Smart way to build momentum and de-risk the program.
And the trial itself, they called it INVEST.
Yep, INVEST stands for in vivo selective expression. It's a phase 1 and phase 2 combined study. First looking at safety and dose, then expanding. They're enrolling adults with B-cell cancers that have come back or stopped responding to other treatments.
And the therapy is INT-2104. That's the lentivector with the CAR targeting CD20, which is on those malignant B cells.
Correct. Patients get a single IV infusion of INT-2104. But here's the really revolutionary part.
No chemo beforehand.
Exactly. No lymphodepleting chemotherapy, no fludarabine, no cyclophosphamide, just the infusion. That's a potential game changer for patients, avoiding those harsh chemo side effects.
That alone is huge. So the moment of truth arrives.
October 2024. The news breaks. Interius has dosed the first patient ever with a durable in vivo CAR gene therapy.
Must have been an incredible moment for the whole team.
Phil Johnson called it a watershed moment, not just for Interius, but for the whole field. And the very early signs were encouraging. The patient seemed to tolerate the infusion well, no immediate signs of severe cytokine release syndrome, and crucially, their B-cell counts started to drop.
Showing the engineered cells were actually working inside the patient.
Precisely, doing what they were designed to do.
I can't even imagine the tension back in Philly waiting for that news. You dose the first human, and then you just wait.
Right. I heard stories that Saar Gill was basically living in his inbox, waiting for updates from Australia. Every day must have felt like a year. This isn't just science, it's people's lives, years of work, hopes, all riding on this.
So when that first positive data came back, B cells going down, no major safety issues?
Yeah, I was told it was incredibly emotional. Tears, relief, a huge validation. All those tough early days, the skepticism, the long hours, it all led to this. It really forged a strong sense of resilience in the company culture going through moments like that.
And that Australian data quickly opened doors elsewhere.
It did. Just a few months later, January 2025, Germany's regulatory agency, the Paul-Ehrlich Institute, gave them the green light to expand InVs into Europe.
That's big. Access to the EU market, and Germany's a key player.
For sure. Phil Johnson pointed out Germany often embraces innovative medicines early, so Interius started setting up trial sites there, aiming to get European patients enrolled. The plan was, and presumably still is, to share more detailed interim safety and maybe some early efficacy data later in 2025.
And what about the US FDA?
They've been keeping the FDA in the loop all along. The plan seems to be to gather enough data from Australia and Europe to really build a strong case before filing the official IND application in the US. In the meantime, they're presenting their work at scientific meetings, sharing the technology, the early findings, building awareness and credibility.
Now, INT-2104 is targeting cancer, but Interius wasn't just focusing on oncology, were they?
No, that's another really exciting part of the story. Their platform is potentially much broader. They were already developing INT-2106 for autoimmune diseases.
How does that work?
Similar idea, but instead of targeting CD20 on cancerous B cells, INT-2106 carries a CAR that targets CD19. CD19 is found on a wider range of B cells, including those that go rogue in autoimmune conditions. The goal is to deplete these harmful B cells and hopefully induce an immune reset.
Like rebooting the immune system to stop attacking itself.
Exactly. Think about diseases like lupus, or maybe multiple sclerosis, conditions where B cells play a key role. Preclinical work looked promising for this kind of reset. It could offer a totally new way to treat these chronic, debilitating diseases, maybe even long-term remission after a single treatment.
That's huge potential. And they had another one, INT-2108.
Yeah, that one's still a bit more mysterious. They haven't revealed much publicly. Given the platform's flexibility, it could be targeting another blood cancer, maybe using a different CAR or even a T-cell receptor, or potentially even aiming at solid tumors, though that's a tougher challenge.
So the platform itself is quite versatile.
Very. You can imagine using these targeted lentivectors to deliver all sorts of things. Different CARs, T-cell receptors, maybe NK cell engagers, perhaps even gene editing tools like CRISPR, although that adds another layer of complexity. The possibilities are really broad.
Which I guess brings us back to the big news from just recently, August 20th, 2025.
Right. Kite, Gilead's cell therapy powerhouse, steps in and announces they're acquiring Interius for $350 million.
That made headlines everywhere. Kite, the king of ex vivo CAR-T buying a leader in vivo.
It's a powerful statement. Kite already has Yescarta and Tecartus, multi-billion-dollar ex vivo therapies. This move clearly shows they see in vivo as the next frontier, or at least a critical part of the future portfolio.
Did they talk about the strategy behind it?
Yeah, Cindy Peretti, who leads Kite, put it very clearly. It's about combining Kite's massive experience in manufacturing, clinical development, and commercialization with Interius's innovative platform. The dream goal, she mentioned, eliminate the need for taking cells out, the complex processing, the harsh chemo. Basically, it makes cell therapy potentially as simple as getting an infusion at an outpatient clinic.
That would change everything for patients.
And absolutely. And Phil Johnson, Interius's CEO, echoed that. He saw the acquisition as the best way to fulfill their mission, getting this technology to patients globally. Leveraging Gilead Kite's infrastructure makes way more sense than trying to build it all from scratch.
And they're keeping the team in Philly.
That's the plan. Establish a center of excellence there, keep the R&D roots where the whole thing started, which is also great news for Philadelphia's biotech, Silicon Valley, as they call it.
Financially, it's a relatively small deal for Gilead, but strategically, huge significance.
Definitely. Analysts see it as Kite future-proofing its leadership in cell therapy. You can't afford not to be playing in the in vivo space if it takes off, like many expect.
So what's next for Interius now as part of Kite?
Well, the immediate focus is the InVs trial. Keep enrolling patients in Australia and Europe, gather that crucial data. We should be looking out for those first real data readouts, probably later this year or early next. If that data looks good, safe, and showing clear signs of efficacy, then you'd expect them to plan for larger, potentially pivotal trials fairly quickly, maybe in the next couple of years.
And success with INT-2104 would boost the whole platform.
Absolutely. Positive results there would pave the way for INT-2106 in autoimmunity, and whatever INT-2108 turns out to be. It validates the entire approach.
And looking beyond Interius Kite, this whole field is really buzzing now, isn't it?
Oh yeah, it's heating up fast. You've got other startups, other academic groups exploring different ways to do in vivo engineering, some using different types of viruses, some using lipid nanoparticles, some using transposons. Everyone's trying to crack the code for safe, effective, direct-to-patient cell engineering. Big Pharma is definitely watching very closely, looking for the winning platforms.
If Interius's lentiviral approach really works safely and effectively in larger trials, could we really see cell therapies delivered like, say, a vaccine or a standard antibody infusion?
That's the ultimate dream. It could democratize access in a way that's hard to even imagine right now. But, there's still big questions. Durability is a key one. How long will these in vivo generated CAR T cells actually last and remain effective? Months, years, we don't know yet. Another is immunogenicity. Will the body eventually mount an immune response against the viral vector itself, preventing repeat dosing if needed? And then there's the solid tumor challenge. Can this approach be adapted to work effectively against solid tumors, which have complex microenvironments that often shut down T-cell attacks? These are the big hurdles the field needs to overcome.
So still a way to go, lots of unknowns.
For sure. But even with those unknowns, what Interius has achieved is already historic. From that initial idea born of frustration to being the first to dose a patient with this kind of durable in vivo CAR T therapy, and now this major acquisition by a field leader, it's an incredible biotech story.
It really is. It perfectly embodies that spirit of innovation, of asking why not, and just persisting through incredible challenges.
This whole story of Interius BioTherapeutics really drives home how sometimes the biggest leaps forward come from challenging the status quo, from asking that simple, radical question, why not do it inside?
It makes you wonder, doesn't it? As we look ahead, what other seemingly impossible medical hurdles could be overcome if we just started to ask seemingly impossible questions? Thank you for joining us for this story on BioTales. Until next time, keep asking those bold questions and stay curious about where science takes us next.
The circuit hums with a heartbeat new, tomorrow's edges in view.